This is what this picture here is. All those lines are actually an inanga ear bone that has been taken out of its head before it goes into a whitebait patty. And I polish it by hand with a piece of sandpaper to reveal all the internal rings. And the ear bones in whitebait and tuna tell us so much about their life. They're diaries. They're called fish diaries. They're really fascinating. So I'm just going to talk about how climate change is affecting Aotearoa's marine and freshwater environment. Next, yeah. Do we know if our freshwater fish in New Zealand are being affected by climate change? I'm going to talk a little bit about a case study about my research that's currently ongoing about how I'm reusing old collections of tuna ear bones to try and reconstruct or look back in time and see how climate change is affecting our tuna. And then some future research directions that we might need to go in to understand the effects of climate change on our freshwater fish. So climate change is happening, and it's a well-known fact that our planet is getting warmer. So last year, the Tasman Sea in New Zealand was 6 degrees warmer than average. And that has a massive effect on our fish populations in terms of their ability to deal with warm temperatures. And this here is a graph showing how extreme that marine heat wave is what it's called. And all that red around New Zealand is basically showing you hotspots of sea surface temperature in New Zealand. And the white is normal temperatures, and the red is alarm bells, 6 degrees above average. We're also seeing more extreme weather events around the world and in New Zealand, more frequent cyclones, extreme rainfall events, drought, storms, all of those kinds of things. And this here is actually just a picture a couple of months ago of the Waihora Bridge between Fox and Franz Josef Glacier, where there was a massive rain event and it took out the bridge. And actually, what scientists are starting to think is that because the sea was so warm at that time, it changed the rainfall pattern in New Zealand, and it resulted in a very extreme event, such as we saw down in the South Island. And then you might look back in time to 1997, 1998, where there was a very extreme drought event in New Zealand. And you can see that brown all along the east coast of both North and South Islands show where rainfall was 20% lower than average. So we are starting to think that these kind of extreme climate events in New Zealand are affecting our taonga freshwater fish species. So these fish here are inanga, they're really beautiful. And there's some indication that changes in the sea level affects their spawning habitat. So like I said, because they lay their eggs on vegetation, riparian vegetation, where the river and the salt water meet, because of changes in sea level rise, we think it might be changing the distribution of inanga spawning habitat in New Zealand. That's just some pictures of the eggs and the spawning habitat that they lay on. Also, these extreme rainfall events that happen during their spawning season can smother the eggs, and it results in high mortality of the eggs, meaning we're not getting a lot of whitebait during the whitebait season. Inanga are also found in Australia, and they're starting to think that those changes in ocean currents, because of changes in temperature and changes in rainfall, are affecting how many inanga make it back into the rivers. So that's happening in Australia. We're not sure what's happening in New Zealand. If we look at other science across the world, we see that climate change is affecting other tuna species. So in Japan, they have a big tuna fishery. And what's happening for the Japanese eel is that changes in the marine environment are affecting the spawning grounds of tuna. Tuna reproduce in the sea, and they're not able to find their way back to their spawning grounds because the conditions have changed so much. And that little picture on the right is actually a larval tuna. That's what they look like in the sea. They're called a leptocephalus. We've never found a long fin larval tuna, never seen one before. and only 9 shortfin have been caught previously, so that's what they look like. Nothing like the tuna you see in the Awa. You can change. And what they're seeing in Japan is that they're getting less and less tuna back into the rivers from the sea. And basically the black line is a climate event called El Niño, La Niña. You might recognize those terms. And the dashed line are the numbers of tuna coming into the rivers. And you can see they follow a cycle of very high and very low, and that tracks with climate change. So this is a little video of some beautiful eels in Lake Wanaka. And we really don't understand how climate change affects New Zealand's eels, and they're very, very special. The longfin eel is actually the longest lived eel in the world. They can live for over 100 years, so they're really, really special. Shortfin eel can live for over 50, so they spend 50 years in the Awa, longfin eel over 100. So if there's any changes or impacts from climate change, they can affect eels quite badly because of how long they live. So we just did a review that was commissioned by Te Waihora recently of an assessment of what do we know about our freshwater fish in New Zealand. And we covered biology and pollution and habitat degradation on all sorts of effects. But the biggest thing, knowledge gap for us, was climate change, which you can see here in this white circle. So we know a little bit about fishing pressure, water quality, predation, but we know nothing about climate change. And one of the reasons is that we don't have long-term data of our freshwater fish. And there are some critical knowledge gaps that are also missing in terms of their basic biology. So I'm just going to explain what fish ear bones are. There's a selection of them here on the right-hand side. So every different fish species has a different shaped ear bone. They're quite beautiful. Sometimes people make jewelry out of them. And this is a picture here of a tuna ear bone being taken out. So you basically make a little cut on the top of its head, and the ear bone lies just behind the brain. So they're made of calcium carbonate, the same material as shells. And fish use them for hearing, for balance, for motion, and for swimming. But they actually have so much information contained in them. This is a tuna egg that was caught in the sea. And the really important thing about the ear bone is that it forms at birth. So as soon as an eel egg is born in the sea, the otolith is formed. And that's it there in the tiny white circle. And they grow continually throughout a fish's life. So they grow from the egg to the larvae, to the juvenile, to the adult. Every single day a ring is put down on the ear bone. So this is what the rings look like. So for a larval eel, we can have an entire time series of its life in the marine environment through the daily rings that they have deposited. Another really special property is that they put down a ring every single year called an annual ring, and that's those dark rings here on the ear bone. And that's what we use to age tuna. We count all of those rings to figure out how old they are. And if you look at the tuna ear bone, it looks quite like a tree. They have very similar properties where they put down a ring every single year. So the distance between the rings, if it's quite wide, it means that the growing conditions have been good. And if the rings are narrow between years, it means the tuna or the tree has had very bad growing conditions. So I'm trying to understand how those changes in the distances between every annual ring might be related to climate events for tuna. So to do this, what I've done is I've kind of raided NIWA's collection of tuna ear bones that we have. And this is a selection of some of them from as far back as 1974, where some of the first studies of tuna were done in New Zealand. And my study site is actually Te Waihora, Lake Ellesmere. This is actually one of the most extensively studied lake populations in the country on tuna. So I've been fortunate to access a lot of ear bones from Lake Ellesmere from different years, 1979, the 80s, through to the 1990s and the 2000s. So there's lots and lots of ear bones available that I can reanalyze. So just to explain how this works, every single ring on a tuna ear bone is associated with a calendar year. So the very last, I'm not sure if you can see that, the very last ring on the outside of the ear bone was formed in 2006. The next one is 2005, 2004, 2003, so on. So you have a time series of its life. And then I measure the distance between each of those annual rings. And I do that for every single tuna. So I've done it for, I don't know, 300 tuna or something. It's a bit time consuming. So then every tuna that has its growth formed in the same year are aligned, and then we get an average of the growth rate in that year. And this is what it looks like. We create a long-term data set of tuna growth rates. And this is basically what it looks like. So it's a 50-year time series of changes in shortfin tuna growth rates. And it's one of the longest that actually exist in the world, because other eel researchers don't tend to study adult tuna, so they don't have a lot of ear bones available for analysis. So basically, the top panel above 0, the positive values, are years when tuna grew really, really well in Te Waihora, and then the blue panel are years when tuna grew really poorly. So we're trying to understand why in some years are they growing really fast and why are they growing really slow. So 1973 and 2003 were 2 years with exceptionally good growth in the lake, and then 1970, 1985 through to 1990 were years when tuna growth rates were really, really bad. So to understand what is driving those patterns of really good and really bad growth, I've selected some environmental variables and I've basically related them to this time series. And I've chosen 2 periods for analysis. That's just a statistical thing. So the two time periods I'm looking at are 1962 to 76 and 95 to 2009. And basically, from 1962 to 1976, there was an effect of El Nino on tuna growth rates in Te Waihora. So when there is a strong El Nino event, eel growth rates decline. And there also was an effect of summer wind. So when it gets really, really windy in the summertime, tuna growth rates get slower. And the exact same pattern was found between 1995 and 2009. When we have an extreme El Nino event, we have declines in shortfin tuna growth rates. And if we actually just have a look and see what kind of significant climate events happened over those time periods. And in 1968, there was a massive storm, Cyclone Giselle, or the Wahine storm, and that changed the conditions in the lake, and it was a very windy period, so that wind relationship with growth rates is probably because of the Wahine storm. And then we have some extreme El Nino events. There was extreme El Nino in 1997-98, like I had shown previously, and also in 1972-73. So we're seeing signatures of these extreme events in our tuna ear bones. So I had a quick look at our tuna earbone archives that we have at NIWA to see if there was anything within Ngāti Kahungumu's rohe that we could look at, perhaps to go back in time and use this type of method. And unfortunately, there wasn't an awful lot of tuna earbones available for reanalysis. But that's not to say there's not opportunities in the future to keep the tuna earbones and to look at this kind of research. So some future research directions. One of the key things is that climate change is not the only pressure on our freshwater fish. There are lots of interacting factors. So we need to understand how they all interact together. How does fishing and climate change and habitat degradation affect our freshwater fish? It's a big question, but that's kind of the direction we really need to go in. We also need to start looking at other rivers and lakes if the same patterns are consistent in other parts of the country. And longfin eel, we don't know what the effects of climate change might be on longfin eel. And then also a key question is, can we apply this method to other species like our inanga, like our kokapu, pātiki? Yeah, and that's it. And I'd also like to just acknowledge Ngāi Tahu for giving their permission to use the ear bones that from Te Waihora. Kia ora. I think someone asked if I was native Indigenous Irish on the question on the Slido. Is there noch me askunte and chlor agus tá kind machanga shunta said Gaelga. So I'm from the west of Ireland and I speak Irish. I grew up in a household that spoke Irish. So yeah, I guess I'm Indigenous.